Treangen from the University of Maryland, who will give us some more insights or let's say things to think about which could for future CAMI challenges.
This is a really lightweight talk, and it's really designed to kind of stimulate discussion more than anything. Very light on results and just some general impressions of detecting pathogens in metagenomic samples after having thought about this problem for a little while. Really in the context of some of the current tools that we could use to do this, and kind of implications of different use cases. And I'll get into that. But so before I start, I want to say there's a lot of focus in it, and it's on kind of names sometimes, and it's important. I just wanted to point out that there's a lot of ways that we can name things. I guess the recommended way is to start by contacting this international committee for help, but the point is there's a lot of names which make it seem when you see a certain microbe or pathogen that they might be very similar, and they are, or names that are different and actually are things that differ by a single gene or very little differences at all. But we have DNA hybridization, some idea of basically 70% value or better, it's a same species, and if it's lower than that, it's not. In silico, kind of the equivalent based on mummer or blast, this 97% average nucleotide cutoff, and then gene presence absence. And this is all to show you that basically things are named differently, consistently differently everywhere. So we have to be careful when we treat things kind of as global bags of k-mers or other global bags of genes or whatever. Sometimes it's very important to know the specific biology of a given bug, of a given microbe or pathogen to be able to call it correctly. And I'll try to emphasize that throughout. But basically this is a plot from this paper in 2006 that kind of shows some of the diversity. On the x-axis we have average nucleotide identity, and then on the y the total DNA sequence that differs. And this is just basically to show, if you stick over on this side of the plot, that there's some definition of a species cutoff where things of the same color are the same, related or same species, and then basically that there's a wide kind of sampling of average nucleotide identity and differential gene content for things that are called or named similar things. So basically, just to show that sometimes it just varies incredibly, and this is why for detection, and again why I think this is potentially a useful angle for future CAMI challenges for focusing on this, because I feel it's really a subset or a special use case of kind of what you might want to get out of a metagenomic sample. If you're really focused on detecting something that is a pathogen, you might want to kind of just forget about names, just completely, and think about what are the pieces of evidence that we need to see to say that this is present in the sample, even if it has a very good match to the database to something that's well known, and which might either create a loss of sensitivity or maybe even false positives. And this is another plot that I borrowed basically for things that we might care about in terms of pathogens or things that we might get very nervous about. Basically, again, on the x-axis, different. We have contagiousness. Off to the right, it's worse, and then deadliness on the y-axis going up. It's worse. So fortunately, there's nothing up here. Otherwise, that would be very bad for everyone. But basically, they get the idea of things that are kind of really important to find are things that are very deadly, such as Ebola, or even and then thinking about things that are very infectious, and thinking about how we can prioritize, or these will come up in different environments, either food contamination, safety, outbreak event, as with Ebola, or maybe even a biodefense or bioterrorism incident event. So basically just to show that there are obviously a lot of things that, a subset of things that it's very important to not necessarily not only know if one of these is in your sample or you could track or detect, but also if anything behaves similar to that, to find it as well. So my kind of, it might be a pathogen if people wear suits like this around you, And that's something where I don't think there's any room for discussion. If there's something that needs to be handled in a very stringent biosafety lab, we want to be able to find confidently in samples for obvious reasons. And of course, this health impact of focusing on things like this, and again, just to give an idea of motivation for focusing on this is, lots of deaths from Mycobacterium tuberculosis each year. Antibiotic resistance is responsible for a growing number of deaths each year. And then think about food contamination, bioterrorism, subways and cruise ships for areas or places that might be interested in detecting these. And then another thing I wanted to throw out there is this idea of false positives versus false negatives. I think Alice had mentioned something like this too. But there's different implications, especially in this arena, where false positives in terms of these pathogens could lead to panic attacks and stress, fear of riding the subway, incorrect maybe treatment, shutdowns of certain environments, and overall reduced confidence in detection methods. And of course, false negatives might have even more severe outcomes, of course. Current approaches for how to detect, there's obviously, I'm not going to go into all of this, but just I'd like to think of it sometimes as a known, kind of unknown in a sample, meaning there's a representation of something in the database that you might be able to use to get an idea, closely dependent or highly dependent on the database and taxonomy, and obviously a lot of the different type of approaches that you can use to detect. And of course, then you have some unknown in the sample. DB may be helpful, probably not. Probably going to have to perform annotation. It's a very complex problem. So kind of breaking it into two different categories in terms of detection of things, maybe either known or unknown. And then I guess the other thing I wanted to say is, should metagenome pathogen detection methods operate on kind of bags of k-mers, genes, operons, pathogenicity islands, replicons? What is the right level, or is it all of the above? Do we want to incorporate all of the evidence that we could possibly have on all these different kind of different layers here to see, to confirm that something, again, of interest is in the sample? So one kind of vignette or case study I just wanted to walk through is the Merothrax. And for those that didn't know, these are anthrax-laden letters that were mailed in September of 2000, between September and October 2001. 5 were actually killed, 17 were infected, and then there was an investigation launched to find out who was doing this, where it came from, et cetera, et cetera. Some colleagues of mine were involved on this, but the point I want to say, this is a very specific use case. Something happens of significant scale. Someone's mailing around letters of a nasty bug. And this triggered a very large-scale investigation that lasted over 7 years, and more than that, this is kind of just a quick overview of what was done, and I'll get into this later slides. But just to very briefly go over this is, everything was thrown into it, where a reference genome was sequenced and assembled. All of the molecular techniques that were available were used, in very fine-grained comparative analysis on the new reference genomes and query genomes extracted from the samples or the letters were assembled and compared to, and then genetic differences were used as forensic evidence to try to figure out what was going on. Okay, so very brief and quick overview, but there was a lot of QC and a lot of very rigorous and expensive steps in here to confirm the presence or for attribution of this, and eventually figure out what was going on. Okay, so basically the need for the kind of high quality reference and the lack of one, but a lot of money, a lot of effort, a lot of very talented, bright people looking at this problem for a long time to resolve this and figure out who was doing it and be able to put in jail. So just lesson one, use cases I think are very important. In practice, this was kind of sky's the limit, just figure it out. Unlimited budget, $1 billion indirect costs, 7-year investigation, public pressure, terror, fear, or uncertainty, and implications of false positive, of course, were huge. You don't want to send the wrong person to jail, of course. So it's a pretty big deal to get this one right, okay? So again, really unlimited resources to do that. Second one, the second example of this is a New York subway study where you have pathogen map study for that sampled multiple sites and multiple classification bidding profiling methods called BA anthracis present and two of the samples, I believe. And that raised a lot of eyebrows because, again, as I just said, we want to be able to do this correctly, and this is a pretty big event if it is present. So simply that it called it present was triggered a lot of others, either via blog post or responses to figure out what's going on. I will show in the next slide, but basically on further investigation from others and I self also looked in this a little bit, but basically there was no, you couldn't rule out, but there was no good evidence based on all of the known markers for BA specific biology that was in the samples. Okay, and furthermore, when I looked into it, so a signature marker-based method, one of them, metaflan, when I looked at marker genes that were kind of being highlighted as reasons for it triggering a BA call, I looked at the markers and it turns out most all of them were actually prophage proteins, which are mobile, jump around, and are non-specific. Okay, so it started to kind of, when you take all of that out and you take out some of the other noise, it turned out there was, again, just a lack of evidence for the call. So again, very sensitive, good method, but sometimes you need to, sensitivity and the false positives are important and have big implications. So it's something that I think it's very important to keep in mind. It's something I think you could, if you set up a challenge to kind of evaluate this, you could see the different behaviors and characteristics of the method, different methods. So another, following off of this, so even getting down more fine-grained in this, can a single nucleotide difference matter? And the answer to that is yes. For Bacillus anthracis and plicar biology, basically this nonsense mutation in all of the known Bacillus anthracis genomes interferes with sporulation, which is a critical component of so the expression interferes with it, so the nonsense mutation enables the sporulation. So it's CAMI and others, I mean, it's been shown to be a very specific marker for diagnostic assays. So basically, every time they've looked, it's all the BA genomes have this at kind of position 5 million in the genome. It's this plicar nonsense mutation, and without it, you can rule out in many cases that it's BA. So that's a single nucleotide difference, and this could be genomes that are 99% similar to each other. But if you miss this one difference, you may be making the wrong call on one side or the other. So that's the kind of resolution that I wanted to start kind of talking about, because and this really gets back into what do we need to get to this point? Is it genome assembly? Is it so I think it really kind of involves all of the above when you're thinking about what are the pieces of evidence that are needed to confidently call BA. So visualized in multiple alignment. These are a bunch of Bacillus anthracis genomes. This is the plicar nonsense mutation. All of the BA genomes have it, and everything else and everything does not. So a quick check for this in the metagenomic sample for the subway showed that they didn't have the BA plicar nonsense mutation. Okay, and then of course there's other pieces of evidence. Very critical components are the plasmid presence. So I just wanted to start thinking about for BA detection, you might want to start thinking about finding that SNP plus the presence of plasmids and et cetera. Another example, again, I'll go through very quickly, is Clostridium botulinum. Has very, very high similarity to other Clostridium genomes, and it's basically determined, it's called Clostridium botulinum based on the presence absence of toxin genes, 18 neurotoxin clusters. You want to know if those are present, and that's what's going to determine that you have, not necessarily the relatedness to the other strains. It's really all about the presence absence of these neurotoxin clusters. Okay, and these are the 18. So you would maybe think about for this specific microbe develop an assay where you can just kind of look for these, and if they're present, you'd have a strong basis for calling Clostridium botulinum. And then to kind of further drive home the point is just the fact that botulinum and sporogenies are kind of all spread out together, so they're equally, it's really the presence of these toxin genes which is going to trigger the naming of it as Clostridium botulinum, not really anything else other than that. So, and global similarity, given that it's a very small difference again, may not capture, may falsely assign it to a sporogenes compared to botulinum. So, and this is a recent study that I participated in, but so for example, if you're interested in finding very specific virus in a large complex metagenomic sample, this is kind of a either a fishing expedition or unknown, or maybe you have some idea of what you're after. A very small genome, 3 segments, an RNA virus. Let's say you want to find this in a sample, speculated to cause fever-like illnesses found in South America. Running some of the methods here, I ran several. You're basically, most of the sample is something else. It's the host background. It's other microbial load, and it turns out 0.03% was assigned. And specifically, initially, when I was running some of the classification tools, 4 reads out of 200 million were classified as orthobunia virus, which made me think, is that enough? That's made me think a lot about what's the threshold for really digging in further. When is it enough to call presence absence? Fortunately, you can use more sensitive methods. You can find many more reads, 1,000 to 2,000. But still, this idea of what is the threshold there? What's the limit of detection in terms of reads that you see in coverage of a certain organism to say certainly it's present, and specifically with respect to viruses which have multiple segments, confirming presence absence of all 3 segments and not and associating them. So and then this idea of no straightforward to link viral segments, but I think it could be another interesting use case for the pathogen detection for a viral example, kind of a needle in a haystack and without a good reference to see how methods do. So just conclude, just possible future detection challenges based on some of the vignettes or examples I highlighted, where you could think about creating kind of mixtures of, maybe a B-series genome plus plasmids and the allele, and will that be enough to trigger a call? Sporogenies plus the neurotoxins, Staph aureus genome with antibiotic-resistant cassette, RNA viruses, and toxin gene plasmid presence absence, and some of the plasmid presence absence we've already talked about, but sometimes it's very important to call all of the replicons to be able to say anything specific about a certain pathogen. So with that, other thoughts or points to consider. Kind of associating genes to replicons is hard and likely requires assembly and assembly graphs. SNPs are often important, adaptation is important, and poorly assembled unavailable host background sequences is common, so it's something that detection methods may have to account for to get rid of the noise per se and focus on the signal. And then just kind of summarizing what I said, for Bacillus anthracis, you could think of maybe this type of approach for saying a very good pathogen detection method that's tuned for Bacillus anthracis might consider the Plicar allele, the 2 plasmids, and the phylogenetic placement. And if it checks the box on all of those, you'd say it's there for sure. And if it doesn't, maybe 2 of the 3, you just don't have confidence in it. And again, if you were to apply this to some of the previous samples that called presence, it would come up as low confidence or not there. So, and again, just to stimulate discussion for future challenges. So I'm definitely interested in hearing any feedback. But just wanted to go through a few examples of things I've looked at or others I've looked at recently in terms of pathogen detection and metagenomic samples. Thank you.
How would you design like a challenge for that? Do you have any ideas?
I've played around with it a little bit, where I've selected certain sets of backgrounds and certain sets of complexity of metagenomic samples that I've simulated, and then you can, if you want to go after the limit of detection angle, you can spike in the pathogen at different abundances, whatever that % of the sample or whatever coverage you'd like, and kind of ladder that up and see when are the methods seeing it or not. And then in terms of presence absence of genes, I've played around with adding and removing toxin genes of interest and seeing if any of the methods would notice or care. Some of the ones that actually have gene databases or plasmid databases do care, and they found, such as LMAT, will actually go and say, I found this gene and I found this plasmid. It won't make any association, but if you saw that, at least you could say, well, I have the chromosome and I have this toxin gene and I have this plasmid, so maybe the potential's there. So just by creating kind of synthetic mixtures of different scenarios, either with or without, to see if you could really confuse them or challenge them to say, is it below their limited detection, or is it beyond what they can do based on just looking at K-mers? So, I have some sample data sets. It's kind of similar to the simulated metagenome framework that CAMI generated. So, but based on that, basically different backgrounds of complexities and different pathogens, throwing them in there, running the classifiers, and seeing what they say based on.
But the tools you are testing, are these sort of general tools, or are they?
I've used general and I've used more specific ones that claim to operate, can offer some type of confidence. Others that just, I'll say that, I'll call it's present if more than 1% of the sample they call as, label as that pathogen. I'll say it's present.
Well,
so,
because I can imagine that tools are very specific for specific pathogens. Just looking at a bunch of genes or something like this. Yeah. And they will, of course, not work for any other.
Oh, that's right. That's right. That's exactly right.
Tool which is like generally applicable.
Yeah.
You could imagine like a challenge for pathogens where participants are actually encouraged to overfit for those pathogens.
Yeah.
Yeah. No. And that's, I guess, kind of my point is I think it's almost necessary. I think the more generic approaches, I think, unless you start layering, I don't know what that would look like, but I think you almost have to fit to the specific biology of the specific bug to say, here's what we know, and see if it's there.
Yes.
And then the unknown, I don't want to get into because that's a even trickier problem. But for that, at least, we should be able to do that well. Be able to say, for a given sample, here's the signs of evidence to say, look, we have confidence or not that this is there, rather than. So yeah, I think that's. And certainly they wouldn't work at all or be very good at more general cases at all.
So I'm working with some people that have a very similar problem in that they've got ancient DNA and they're interested in finding a pathogen in this ancient DNA. So it's quite exciting because it's a tooth of it. They're trying to find Salmonella typhus. And the way they're doing it, which seems to me reasonable, is just trying to determine whether by mapping to the reference genome, you've got a consistent coverage across the whole reference genome.
Yeah, and that's usually one of the checks that I didn't throw in that we do. Just a standard. Is it most of the genome covered by whatever that threshold is, 95% of it covered by read mapping? I would argue on that, though, you could have something novel in your sample that won't map to the reference that might actually affect what you're seeing, either the addition of it.
They can't. You're right. They can't get novel diversity in this historic organism. They can only put it in the context of
right. So that's fine for that organism. If that's what, and I think a lot of the cases, and for this, some of the examples I highlighted, a simple mapping-based approach is sure there's consistent coverage across it, gets you a long way. I think sometimes there are issues either in terms of repetitive sequences or single regions that are very important that you might be able to not get a very clean SNP call out of, which might make it more complex. But certainly, I think that's a start. I've seen some issues. Part of that is has been thresholding of what is how much of the genome do we need to see mapped to call it present. Obviously, when I've done it, it's like 99% it's there. But what if it's 61%, for example? Is it there or is it not there? Because there might be some sequencing artifacts that would, especially with htDNA, the fragmented DNA, you might not get a full representation of the genome. So that's what I'm kind of thinking about. And so in the case where you have a great sample and it's fully covered, problem solved. If you don't, what's that look like, and how much do you need to see before you convince yourself? And that's where I kind of try to bring in this idea of unique signatures or marker-based approaches. If you cover those, it's there maybe, but there could be other biology going around too that you're missing if you focus on those. So I think it's really microbe-specific. For that, it might work great. For others, there might be some other presence absence of certain genes that could really affect things.
And then as a secondary question, why don't they remove all the prophase genes from metafanding?
So that's it was all computationally.
I know, but having done I know how they do the computations. They're looking for unique markers based on that. having done that, then they could just remove
Yeah,
the protein.
yeah.
That does seem like a very stupid thing to have in your...
Yeah, I saw that and I was surprised. But yeah, that's why annotation is important. So if you know that, you just get rid of them, and that's what I think that was the standard practice for a lot of that stuff, where you just remove because they can be non-informative, and a lot of times for that, it turns out a lot of the unique stuff are these mobile elements, just because the DB is undersampled. So
I think that could be an example of where it's not so much the tool developer as how the tool's applied, because Nicholas Agata, the device maker lab, actually spoke to the Subway metagenome people before they released their paper.
Yeah.
And he said there isn't evidence.
Yeah. Yeah. No, that's true. And
they said, well, your program said there is, so we're going to, actually, misinterpret.
So again, I think that actually feeds into my discussion of detection methods. So if you were trained or tuned to be a BA, you can run that. Because again, I think what Nicola and I talked to him as well is, if you look at it, it's not saying that, but you can say, oh, it's lighting up that. But if you really wanted to make that call, if the use case was that level of threshold, you would throw everything at it that you had to confirm that. So you wouldn't just go off and say. So I agree. Yeah.
What do you do about contamination? Because when you've got those low numbers of reads, even if your computational methods are quite good, there's always the concern that that low level just represents some spillover from another sample or other sequence.
Yeah, I can talk about my personal experience in a laboratory where we took extreme measures of control for sample control and signature control. So each one had a barcode, and we knew exactly all the reads that were generated from previous runs. So you could control for that. Okay. Now in terms of contamination, there could be something in there that either false positives or something else that gets in there that you could say, what is this? And sure. That's an issue. And again, I think it just depends on what the use case is. But contamination in terms of isolate genome assembly or whatever, you can remove for control. For this case, where it's such a small fraction and anything could look like a potential signal, you certainly could overcall things even in that instance, because either the threshold again is so low that you'll start everything looks like it's present or contaminated. But in terms of overflow between runs, there's ways you can control for this. I think pretty precisely if you're very, very careful about this in these environments, where you can make sure that previous sequence runs are not bleeding over into your current sample. So that's from experience, I say that's pretty much a control problem. If you take the necessary steps and invest in all the necessary precautions and spend a lot of money. So
we have samples which are sequenced elsewhere, like in Africa.
Right? Yeah. You're right. So I agree. It's a problem. In a controlled environment, obviously, it's much easier. So. Not really sure what else to comment on that.
Yeah, more questions? Well then, thanks again.
Yep.